Welcome to the latest Deep Evolution news. In this installment, roots of Cambrian Explosion exposed in new Ediacaran biota uncovered in China, and a promising option for life on Saturn's moon Titan was dealt a severe blow. Let's dive into it right away. First of all, we may finally have confirmed how far back in time the so-called Cambrian Explosion actually stretched. The Cambrian is the first period of the Phanerozoic, which is the eon lasting up until today, covering the time of the advanced multicellular animal and plant life that we are more familiar with. We already knew that the explosive nature of this event has been slightly overstated. The problem is just that the fossil record from then is simply quite spotty, with a lot of gaps intermitting the few occurrences on which we can pin the presence of a handful of animal groups. Genetic analyses already did place the actual diversification of Bilateria and the origin of the different known animal phyla much earlier than the Cambrian, well into the preceding Ediacaran period. However, fossil evidence of these ghost lineages is very scant and often dubious. The Ediacaran fossil record is dominated by both macroalgae and quaint life forms that are presumably animal but cannot be confidently linked to the later Phanerozoic animal phyla, even though many attempts have been made. The current consensus is that these constitute one or more separate radiations, often called the Vendobionta, and that have since gone extinct. Vendobionts largely appear to be sessile or stationary animals, with some exceptions such as Dickinsonia that seems to have left traces on the sea bottom in some cases. Trace fossils do occur in the Ediacaran already, but not with the same intensity and complexity as we see from the earliest Cambrian. Apart from a few trace fossils, there's limited unambiguous evidence of bilaterians living during this period, with some notable exceptions such as Kimberella that, however, are hard to link to specific animal phyla. A recent paper on the JiangChuan biota from southwest China seems to showcase a notable number of bilaterians actually coexisting with typical Vendobionts. There are at least 3 genera of Vendobionts distinguishable, namely Elienum, Cycliomedusa and Lobodiscus. Among recognizable animals that are not bilaterian, we find presumed jellyfish-like cnidarians such as Conomedusitis and Hautia. One form, called Ctenorhabdolus, seems to be a ctenophore, a group including modern-day comb jellies. Among the apparent bilaterians they found were the following: a large number of worm-like or vermiform animals, of which the biggest one is about 6 cm long and 2 cm wide. A slightly different, more sausage-shaped vermiform fossil with a mouth-like structure. And even more exciting, there are a number of fossils very similar to the Cambrian Heterogaster, which is considered an ambulocrarian. Ambulocrarians encompass both echinoderms, such as starfish and sea urchins, and the hemichordates, such as acornworms, and is the sister group to the chordates, which includes vertebrates. There's even another ambulocrarian found that can be identified as the Cambrian Phlogitis, as well as the infilling of a dwelling tube of another hemichordate named Margaratia. So our closest, most ancient relatives are well represented here as well. The many elongate vermiform fossils are also interesting because many of them display what seems to be an extensible proboscis at the mouth end, presumably for grabbing food. So this immediately evokes an association with the proboscis worms, also known as priapulids, that are a hallmark of the Cambrian, where they were very numerous. Priapulids belong to the greater group of ecdysozoans, to which also arthropods belong. However, there are some important distinctions that prevent a confident alignment with this group. First of all, there are no traces of circumoral teeth on or around the proboscis, the most telltale sign of a priapulid. Second of all, the most unusual structure of these vermiforms is the presence of an anchoring structure at the opposite end, which doesn't quite make sense for a burrowing animal like a priapulid. The authors also won't go as far as to giving a definite classification, but given the musculature needed for such a proboscis, they conclude it must be bilaterian, albeit an unusual one. It is presumed that the anchoring structure was needed to provide holdfast under marine conditions that are different from modern or even early Phanerozoic ones. All in all, these finds are an interesting addition to piecing together how animal life rose into existence. Keep an eye out for a future deep evolution dive video of mine, where I will try to reconstruct what we know of the different stages of this succession towards the Cambrian explosion. An interesting idea that opened up for the possibility of life even in deep freezing temperatures such as those on Saturn's moon Titan has unfortunately received a severe blow. In 2015, a team of researchers from Cornell University posited that seas of liquid methane, such as those that exist on Titan, could potentially allow for the formation of membrane-like structures. These could form the basis for hypothetical cell-like structures termed Azotosomes, and thus simple life forms could persist even under freezing conditions using liquid methane as a medium instead of water. The membranes would be made from a substance called acrylonitrile, which under Earth conditions is a colorless, volatile liquid that smells a bit like garlic or onions. This compound, used on Earth in the manufacturing of synthetic materials, has been detected in the atmosphere of Titan already, so it is a promising candidate for any xenobiological life there. The authors of a new study put this to the test and recreated Titan's environment to see if any membranes or even azotosomes would spontaneously form there. They mixed acrylonitrile with both pure methane and with pure ethane, both known to exist in liquid form on Titan, and then slowly heated the samples to simulate evaporation, though still at temperatures hundreds of degrees below zero. Not only were there no membranes formed in the methane mixture, more damningly, the ethane formed co-crystals with the acrylonitrile, locking it away for any further usefulness. Since we always would expect at least some amounts of ethane to exist in Titan-like conditions, these would be expected to leach away any acrylonitrile that may exist, and thus no membranes could ever be formed from this compound. This doesn't mean that life is impossible on Titan, just that it cannot be based on this particular substance. Alright, that's it for this time around. Please keep an eye on my channel for the next video. Subscribe if you haven't already and hit the bell icon so you don't miss anything. For now, stay sharp and stay evolved.